Hi everyone, we're doing phospholipid synthesis. We've already covered it, but right now we're going to do it in detail, and whatever is left out, that will be covered in today's video. So phospholipid synthesis ये होती कहाँ है? This primarily takes place in the smooth endoplasmic reticulum, and after this, once the phospholipids are made here, they go to Golgi apparatus or Golgi apparatus में इनकी packaging होती है, and then they are sent to be delivered either to the membrane of the organelles या तो membrane of the entire cell या तो there are exocytose out of the cell, right? And why are the exocytose? लेकिन ये तो phospholipids हैं. You may be wondering कि ये तो cell membrane में होना चाहिए, तो either cell membrane of the organelle or cell membrane of the cell. Why are the exocytose? But remember, if you can recall, मैंने आपको काफ़ी चीज़ें बताई थी जैसे कार्डियोलिपिन, which is antigenic, which has antigenic properties. फिर मैं आपको ये plasminogens के भी बताया था. Then I also told you about platelets activating factors, right? All of these, so they need to be exocytosed. ठीक है, तो इसलिए they're also exocytosed. Now moving forward, सबसे पहले we have phosphatidic acid, and this is a precursor of the all the other glycerophospholipids. तो बाकी जितने भी glycerophospholipids हैं, we have glycerophospholipids and sphingophospholipids. तो बाकी जितने भी glycerophospholipids हैं, उनका जो precursor है वो phosphatidic acid है, and you already know वो किस तरह बनता है, glycerol 3 phosphate, and then you have lysophosphatidc acid and then phosphatidic acid. अब this very important thing कि हमारे all the cells of the body, they all do phospholipid synthesis, all of them except RBCs. बस जो mature erythrocytes होते हैं ना, they do not do phospholipid synthesis. उसके अलावा सारे cells in the body, they do phospholipid synthesis. um, Unlike tag, अब tag जो synthesis थी, remember if you can recall from previous lectures कि tag synthesis सिर्फ और सिर्फ होती है adipose tissues में होती है, liver में होती है, and very important lactating mammary glands में भी होती है, right? So this is the function, these are the different spots where tags are synthesized, और जो phospholipids है ये पूरी body में synthesize होते हैं except RBCs, ठीक है? अब moving on towards phosphatidylcholine or phosphatidylcholine ethanol amine. Choline is lecithin and ethanol amine is cephalin. These are most abundant phospholipids, है ना? Phospholipids, glycerophospholipids and sphingophospholipids combined. सबसे ज़्यादा abundant कौन से हैं? Choline और ethanol amine वाले जो हमारे phospholipids हैं. In other words, cephalin and lecithin. And these are also neutral phospholipids. So you can just say PC and PE. PE is phosphatidyl ethanol amine. And then phosphatidylcholine is PC, right? Lecithin and cephalin. Now, when you talk about their synthesis, तो इनकी synthesis कैसे होती है? सबसे पहले तो आपको nitrogenous base मिलता है, right? The pre-existing nitrogenous base that's there. उसकी phosphorylation होती है, right? And after the phosphorylation, what happens is that they get activated. They're converted to their activated form. तो बहुत simple है इनकी मतलब formation. और उसके बाद they combine with the DAG, which is phosphatidic acid से आ रहा है ना, तो ये इस DAG के साथ combine करेंगे, and finally you will get your phospholipid, right? Glycerophospholipid. अब this very important question about choline कि choline की reutilization होती है. Now what does this mean? अब मैं choline के बारे में I'm going to tell you something very important. Choline को हम कहते हैं essential dietary nutrient, and essential dietary nutrient. But there's another contradictory thing about this कि our body makes this de novo. तो de novo synthesis करती है हमारी body इसकी, ठीक है, choline की. अब you may be wondering कि अच्छा अगर इसकी de novo synthesis हो रही है body में, then how come it's an essential dietary nutrient? जबकि essential dietary nutrients तो सिर्फ वही होते हैं ना जो body में नहीं बन रहे होते, आपको बाहर डाइट से खाने होते हैं. हाँ, इसकी de novo synthesis हो रही होती है body में, but it's not up to the mark, or it's not sufficient. The amount that's made, that's insufficient. So then in order to meet the needs, it's essential for us to even have it in our diet. तो जो कोलिन की जो डेफिशिएंसी है ना, इट्स प्रीटी रेयर. इसकी डेफिशिएंसी काफ़ी रेयर है, इतनी होती नहीं है, लेकिन अगर हो जाए तो मसल डैमेज हो सकता है, मसल डैमेज हो सकता है, और दूसरी वेरी इंपॉर्टेंट थिंग जो कि है नॉन अल्कोहलिक फैटी लिवर डिजीज. अब फैटी लिवर डिजीज, अब नॉर्मली हमें यही पता है कि अल्कोहल की वजह से फैटी लिवर हो सकता है. सो इफ दे आस्क यू व्हाट इज द अदर कंडीशन जिसमें फैटी लिवर डिजीज हो सकता है विदाउट अल्कोहल कंजम्पशन, सो दैट इज कोलिन डेफिशिएंसी. तो एक तो मसल डैमेज कर देगा कोलिन डेफिशिएंसी, और दूसरा नॉन अल्कोहलिक फैटी लिवर डिजीज कर देगा, ठीक है. तो हालांकि इसकी डेफिशिएंसी इज रेयर, राइट? आई हैव ऑलरेडी क्लैरिफाइड दैट. अब मूविंग फॉरवर्ड, there's a very important thing that I'm going to tell you, which is about surfactant. If you can recall surfactant, so what is surfactant? Surfactant basically decreases the surface tension at the lining of your alveoli. है ना? वो जो surface tension है उसको कम करता है, right? And other than that, it also reduces the pressure which is needed to reinflate the alveoli. I mean, if it decreases the surface tension, so simultaneously the pressure which is needed to reinflate the alveoli, that will be decreased. जिसकी वजह से what does surfactant prevent? It prevents a very important term, एटेलेक्टेसिस। एटेलेक्टेसिस को प्रिवेंट करता है। इसका क्या मतलब होता है? alveolar collapse. So it basically prevents what is surfactant? It decreases the surface tension and therefore it prevents atelectasis, which is alveolar collapse. Now you may be wondering, why am I talking about surfactant out of nowhere? So it's important to tell you that when you are talking about phosphatidylcholine, the phosphatidylcholine is one of the constituents of surfactant. अब phosphatidylcholine कैसे surfactant का constituent है? If you pay attention at the clinical name of your surfactant, it is DPPC, which is dipalmitoyl phosphatidylcholine. Dipalmitoyl phosphatidylcholine, or you can say dipalmitoyl lecithin. सो बेसिकली ये हमारा सर्फेक्टेंट है जिसके अंदर डायपाल्मिटॉइल फॉस्फेटिडाइल कोलिन है. व्हाट डज दिस मीन? दिस मीन्स कि इसके जो कार्बन 1 है और कार्बन 2 है, फॉर एग्जांपल ग्लिसरॉल है ना, राइट? सो ग्लिसरॉल का जो कार्बन 1 है और कार्बन 2 है, इन दोनों पर एक फैटी एसिड लगा है जो जिसका क्या नाम है? पाल्मिटेट. दिस इज अ लॉन्ग चेन फैटी एसिड. तो इसके palmitate. So इसके फर्स्ट और सेकंड कार्बन पर palmitate लगा है. Therefore dipalmitoyl phosphatidylcholine और dipalmitoyl lecithin. तो ये हमारा स्ट्रक्चर है surfactant का. So that's the reason phosphatidylcholine is important, because basically वो है phosphatidylcholine ही, लेकिन उसके दो फर्स्ट कार्बन पर palmitate लगा है. So this is surfactant और मैंने आपको उसका काम भी बता दिया. surface tension को decrease करता है and therefore it reduces the pressure that's needed to reinflate the alveoli. जिसके से it decreases the chances of atelectasis, which is alveolar collapse. Now there's another very important clinical correlate जो कि है about fetal lung maturity. So in order to assess the fetal lung maturity, what you need to do is you need to check the ratio between lecithin and sphingomyelin. So lecithin or sphingomyelin. Now, what is sphingomyelin? Okay. Now, before you question me about sphingomyelin, so remember when we were studying phospholipids classification, so I told you there's sphingophospholipid and there's glycerophospholipid. तो sphingophospholipid में you have sphingomyelin और glycerophospholipid में हमारे पास lecithin है जो हमने अभी भी पढ़ा है, which is phosphatidylcholine है ना. तो इन दोनों का जब रेशियो लेंगे, so that will tell you about the lung maturity. But how so? अगर ये value is greater than or equal to 2, it means that the fetal lungs are mature, right? So this is an evidence of maturity. So this is a very important clinical correlate that fetal lung maturity can be assessed by taking the ratio between lecithin and sphingomyelin. Lecithin and sphingomyelin, phosphatidylcholine and sphingomyelin. Now, how would you get this ratio? So you need to take basically some aspiration of amniotic fluid. The amniotic fluid, when you get it, so you can check it up. So this is one of the clinical correlates. Now, also one more very important thing about lung maturity. Now, since we're talking about fetal lung maturity, so we cannot proceed the lecture without mentioning RDS. I'm sure a lot of you know that RDS is respiratory distress syndrome. Distress syndrome. So, इसमें usually what happens is that the baby goes in distress because of the lungs. And why? Because of the lungs. This is again linked with um, surfactant, right, and alveoli. So basically, in respiratory distress syndrome, what happens is that the surfactant levels are very low. And why are they low? Usually, this happens in babies who are of preterm, right? So they are born before it's their due date. So what happens is that the surfactant is very low. There's insufficient surfactant, because of which obviously their lungs aren't working properly, and there can be neonatal death. Neonatal death is death at young age. So what is the treatment for this, or what is something that you could do in order to prevent it? So the first thing in order to prevent it is that the mothers who are expecting, they need to start taking glucocorticoids. So glucocorticoids shortly before delivery, if they start taking it, it will be enough to induce expression of specific genes that cause the production of surfactant. So glucocorticoids, what do they do? They cause expression of specific genes that are there to produce surfactant. The other thing is postnatal admission of postnatal admission of artificial, which is synthetic or natural, administration of artificial or synthetic surfactant. So these are the two things that could be done. Okay, so sorry for illegible writing, but I'm really in a hurry to, write and explain at the same time so that there's no pause, and I am not really good at multitasking. Anyways, the first is glucocorticoids to the mother as they're approaching the date of delivery. The second is, which is postnatal treatment, that is postnatal admission of artificial or synthetic surfactants, right? Now, other than this, this was respiratory distress syndrome, which was in babies, right? Also, you may be wondering how can you, administer surfactant. So that is done by intratracheal instillation. It's done by intratracheal instillation. So it's like sent into the trachea. Now, this is RDS for fetals for the babies, right? Or fetuses. Now, when we talk about in adults, so they normally have acute RDS. Now, in this, what happens? This can affect all sorts of age groups, and obviously, there is alveolar damage, and that could be due to any of the reasons. Maybe it could be infection, it could be injury, it could be aspiration, could cause some sort of, when they're taking fluid from the lungs, so during that aspiration also there could be some sort of insult or injury to the alveoli, because of which the fluid can accumulate in alveoli. So that accumulation of fluid is also known as acute RDS, and this is seen in people of all age groups. So when there is accumulation of fluid in alveoli, what do you think, what will happen? There'll be what will this do? This will impede the exchange of oxygen and carbon dioxide, right? So yeah, this is what happens. So now I've told you about two things. One is acute RDS and one is the RDS that occurs in the babies. And then also you know about the maturity, fetal lung maturity test, and you know about surfactant, which is dipalmitoyl phosphatidylcholine, right? Surfactant, that's its clinical name. Now moving on. When you talk about phosphatidylcholine synthesis from phosphatidyl serine. So that reaction is here. You can see this is phosphatidylserine. So how would you produce phosphatidylcholine from here? First of all, decarboxylation will happen, and after decarboxylation, methylation will happen with the help of S-adenosyl methionine, which is methyl donor, and you got phosphatidylcholine. Now, how do you make phosphatidylserine from phosphatidylethanol This reaction is very simple and very unproblematic. You just have to there should be just a base exchange reaction where ethanol amine of phosphatidyl ethanol amine is exchanged for free serine. So that's it, you got this as well. Now moving on, discussing phosphatidyl inositol. This is super important. Now this is synthesized how? It's synthesized by free inositol and CDP DAG, which you get from phosphatidic acid. Right? So basically what you need to remember, CDP DAG and inositol. Now what exactly happens is that okay, before that, phosphatidyl inositol, and then you get phosphatidyl inositol, right? Phosphatidyl inositol it's a very unusual phospholipid. And in what sense? First of all, it has negative charge, right? And then the unusual thing is that on its carbon 1, you have stearic acid, and on its carbon 2, you have arachidonic acid. Now, this serves as a huge reservoir for arachidonic acid. Therefore, due to that, this is the reason why this entire phosphatidyl inositol phospholipid it serves as a substrate for prostaglandin synthesis. So, in prostaglandin synthesis, what is the substrate? Phosphatidyl inositol. So you shouldn't forget that, right? Why? Because on its carbon one it has stearic acid, but that has nothing to do with us. But the second carbon, which is arachidonic acid, so it's a reservoir for arachidonic acid, and that is the substrate for your prostaglandin synthesis. Overall, this compound has negative charge. So what were neutral? Phosphatidylcholine and lecithin were um, phosphatidylcholine and phosphatidylethaноlamine were neutral. Now moving on, the phosphatidyl inositol it has a very major role in signal transduction. Now you may be wondering how does it do signal transduction? So you can remember G protein coupled receptor. So this is G protein coupled receptor with its 7 transmembrane helical heptahelical. It is heptahelical in shape, right? So it has 7 alpha helices. This is the G protein coupled receptor. So this is the G protein and it is coupled with a receptor. Now outside a hormone comes and binds here, and then because of this hormone binding here, there's some conformational changes here, and initially GDP was residing here, but because of hormone binding here and conformational changes here, that GDP dissociates and GTP comes and binds here, and it binds with the alpha subunit to be precise. And this entire G protein in your phospholipase system is GQ. Normally, in the other, camp cycle, we had what? We had our GQ. Instead of GQ, we had GS and GI, which was stimulatory and inhibitory. Here you have GQ. So what will happen after some time is that this is going to dissociate. Alpha subunit is going to take the GTP and they'll dissociate and they'll go to a very important enzyme, phospholipase. Now, phospholipase is going to do what? Phospholipase C is going to break down phosphatidyl inositol bisphosphate. Phosphatidyl inositol bisphosphate. This is basically, the book has used one term for this, which is polyphosphoinositides, right? So example of polyphosphoinositides is phosphatidyl inositol bisphosphate. It converts phospholipase C converts it to IP3 and DAG. Now DAG is basically hydrophobic, so it remains in your membrane. It comes and it remains in the membrane, and not just remains in the membrane, but it also recruits protein kinase C. It recruits protein kinase C to work for it. Meanwhile, what does IP3 do? Inositol 1,4,5 triphosphate, and this is phosphatidyl um, inositol 4,5 bisphosphate. So what does inositol IP3 do? It goes to the endoplasmic reticulum, the smooth endoplasmic reticulum, and there it opens these IP3 gated channels. Once these channels open, there's a calcium efflux and it moves out into the cytosol. Once there is enough calcium in the cytosol, calcium and DAG, they stimulate the protein kinase C. And protein kinase C, it phosphorylates protein. And when the proteins are phosphorylated, that mediates the cellular response to the hormone. So phosphatidyl inositol, it plays a very important role in the signal transduction. So this is one function of your phosphatidyl inositol. What is its one function? To mediate cellular response, or you could say signal transduction. That's its one role. The second important role, transduction. The second important role of your phosphatidyl inositol is that it acts like a anchor. It acts like an anchor, GPI anchor. Now, what does this mean? GPI stands for glycosyl phosphatidyl inositol. So, glycosyl phosphatidyl inositol anchor. Now, what exactly happens here is that, is there any diagram? Yes, here. So basically, this is a diagram that I'm referring to, but I'll explain to you first. So basically, what happens is that this is your cell membrane, right? And your cell membrane, obviously, what does it have? It has phosphatidyl inositol. Now, this phosphatidyl inositol, with it form, there is a carbohydrate bridge that intermediates, and this carbohydrate bridge, it tries keeping up a protein here. So basically, we are holding a protein with the help of this phosphatidyl inositol. So phosphatidyl inositol also has a role in membrane protein anchoring. So it has a role in membrane protein anchoring, that has given it a name of GPI anchor, which is glycosyl phosphatidyl inositol anchor. So basically, this is anchoring this protein, right? And now you may be questioning, what's the reason for this? What is the reason for this protein to be hanging there in the air? Why isn't it embedded into, or integrated into the cell membrane? So because of it hanging in like this basically allows the GPI anchor protein to have some sort of mobility. So they're like free, and they can be easily, they're just mobile there, so they have some sort of mobility to themselves, and I'll explain exactly what is the role. Before that, specific proteins, they, again, I'm just reading off some lines from the book so that it's clear. Is it mentioned here? No, it's not mentioned here, but I'll let you know. So basically, um, specific proteins can be covalently attached through a carbohydrate bridge to a membrane bound phosphatidyl inositol. And what is the example? One of the prime examples of this is lipoprotein lipase, right? Lipoprotein lipase, I'm sure you must have heard. And when I was teaching the topic, I remember telling you guys repeatedly that you have your blood vessels, and in blood vessels, you have lipoprotein lipase. It's there, right? You may be wondering that how. I'm sure you never wondered, but okay. Let's put it that way. Imagine you were wondering that how is that lipoprotein lipase there in first place? Why isn't it? And I remember using the word walls. So it's attached to the walls of your endothelial lining, right, or the capillaries. But why doesn't it flow with the blood? That's because there's something that's holding it. And what is that something? Carbohydrate bridge, and then finally phosphatidyl inositol. So our GPI is there to protect it. So again, lipoprotein lipase, it's an enzyme that degrades talc in lipoprotein particles, is attached to capillary endothelial cells by a glycosyl phosphatidyl inositol anchor, glycosyl phosphatidyl inositol anchor. Now, also moving forward, being attached to a membrane rather than being integrated or being an integral part of the membrane, this allows GPI anchored protein increase lateral mobility on the extracellular surface of the plasma membrane. The protein, it can be cleaved from its anchor by the action of phospholipase C. So how can this protein be cleaved? It can be cleaved with the action of phospholipase C. So if you can recall, there was phospholipase that was cleaving this right bridge and releasing lipoprotein lipase. Now, if there in because of any reason, if there is deficiency in the synthesis of GPI, which is the anchor glycosyl phosphatidyl inositol, then this is a clinical condition that I'm going to explain to you right now. So if there's deficiency of GPI, what happens exactly in hematopoietic cells? There is a disease, a hemolytic disease develops that has the name paroxysmal nocturnal hemoglobinuria. So basically, GPI anchored proteins, they protect blood cells from immune system. Now you have blood cells. Now blood cells, they could be readily attacked by your white blood cells, but because there's some sort of anchored proteins, GPI has anchored some sort of proteins. Thanks to GPI, that it has anchored some sort of proteins, so the body detects those proteins and it doesn't attack on those cells, RBCs. So they are safe and secure from your immune cells. So again, but what happens if there's deficiency or lack of this GPI? Yes, you might have guessed that our body, it doesn't any longer recognizes RBCs as self, and it starts attacking on it, due to which what happens is that we have hemolytic uh, disease, which is paroxysmal nocturnal hemoglobinuria. So again, some of the GPI anchored proteins protect blood cells from the immune system, which recognizes foreign substances such as viruses and bacteria in the body. The lack of GPI anchored proteins on the red blood cells are no longer recognized as self and are susceptible to destruction by complement mediated lysis. So this is the second very important role, that it helps protein anchoring. And see, I told you the lipoprotein lipase, I've already told you, right? And the second very important thing it was doing was it was protecting our RBCs. So lipoprotein lipase was there, but also RBCs, it was protecting them. So enough of phosphatidyl inositol. Now we know how useful it is. It's super useful in mediating cellular signals and helping with the. It helps with the cellular regulation and all of that. What do you call it? Transduction, signal transduction. So it helps with the signal transduction, but it also helps, anchoring the proteins to the membrane, like lipoprotein lipase, RBCs. But we're not just done. I thought we were done, but actually, I think I just forgot this diagram. So very important. You can see this is your cell membrane. This is the lipophilic side. Chains of phosphatidyl inositol, they're inserted into the lipid core of the cell. So this is your phosphatidyl inositol, which is embedded in the bed in the cellular membrane. And then what happens is that I told you there's a glucose bridge, right? So this is your carbohydrate bridge that is helping to anchor a protein. So this is the anchored protein. Now this glucose bridge, it is made up of, what is it called? It's called glucosamine. See, glucosamine is the glucose bridge that's right here. That is helping to anchor the protein. Now, when you talk about glucosamine and all the anchored proteins, what comes to your mind? So let's see this extra information. GPI anchor is a glycolipid that actually it's not a glycolipid, but I found this on internet, but this is phospholipid, right? And phospholipid, that too glycerophospholipid, and glycolipid is not a category of your glycolipid is basically glycosphingolipid, remember? So anyways, but if internet says so, maybe it can be true as well. I'm not very sure about that, but okay. So it attaches certain proteins to outer surface of the cell membrane to keep them stable and correctly positioned. Now, these are the five examples of GPI anchored membrane proteins. Now, one of them we know lipoprotein lipase, and the other we know is on the RBCs. But other than that, you have alkaline phosphatase, which are anchored in the intestinal and placental membrane. Then you have acetylcholinesterase, which is at the neuromuscular junction, CD5, and you have CD59, prion protein. I remember studying this. Then you have 5-nucleotidase, folate receptor, urokinase receptor. So these are some examples of the GPI anchored membrane proteins. What about glucosamine? What is it? It acts as a key sugar that connects GPI core to the phosphatidyl inositol So it connects GPI core to the phosphatidyl inositol helping form the bridge between the protein and the membrane. So it helps form the bridge between the protein and the membrane.